Good evening, everyone, and it's great to be speaking to members of the Team Valley Geological Society again. Stromatolites are layered rocks created by microbes interacting with their environment. And you can see on this slide on the left what looks like a fossil hand. So I've said this is the course of future evolution foretold by these stromatolites, because that's a Precambrian stromatolite well before humans are old. Yeah. You can find stromatolites today and in the geological record of the last few 100 million years, but they're particularly abundant in the oldest part of the record, the Precambrian. A rich and diverse Precambrian fossil record has now been discovered, most of it microscopic. Stromatolites are by far the most common visible evidence of life, and yet they are not simply fossils. Now, what I'm going to do now is I'm just going to pause the PowerPoint. I'm going to show you, instead, I'm going to show you a little video clip. It's on YouTube, and it's called "What Are Stromatolites?" So you can look it up yourself if you have the inclination to do so at any point. So here we go. It's a nice Australian lady talking.
Can't hear it, Ian. What's wrong with that?
Hello, I'm just pausing the video. You're saying you can't hear?
For me, the sound is completely broken up and can't hear what
hear
you're
it,
saying
Ian,
at all. No.
Oh, right. Okay. That's funny, isn't it? Because it's playing through my loudspeakers. So I thought it would be just as you could hear it just as well as I could.
No.
Oh, right. Okay. In that case, I think it's probably better just to omit that video. So it's called "What Are Stromatolites?" and it takes you through. You can see there's some pictures there of modern ones. It shows you the microbes that are inside them. It shows you Shark Bay, which I'm going to talk about in the talk. And then it talks about the importance of photosynthesis and how photosynthesis was really important in generating oxygen early in the history of the Earth, and how stromatolites have been around for a very long time, and therefore we've got to look after them like we have the rest of creation. So it's just a 2-minute video, and it's really quite a nice sort of succinct summary. But because of that technical issue, we'll leave that, and I'll go back to here. So and I'm going to hide the fluid meeting controls. There we are. Right. Okay. Now then, in the first slide just now, I showed a photograph from the Isle of Islay, and this was where I first encountered stromatolites. In 2016, I found some examples at a different level than those I'd previously looked at, and I found it on the land of the Fletcher family at the farm Perseus, bed and breakfast at very reasonable rates. I produced this leaf. I thought you were supposed to be muted, John.
I'm not. No, I retain control.
Okay, right. I produced this leaflet for them to explain the stromatolite outcrops on their land. And there's you can see the outcrops on the right, but it wasn't feasible to sample them. Then in spring last year, I spotted a stromatolitic boulder in a pile of field stones when I was staying at Perseus, and Donald Fletcher agreed for me to take it away to be sawn, and I said I would return with half of it for his family. It was too big for any of the normal saws I use, so I went to a monumental stonemason. I recommend them in Callow End. It is being sawn today, and I'll show you the sawn rock at the end of the talk. So that's the rock of the month, if you like. Okay, on with the talk about stromatolites. In Precambrian carbonate successions, we see very many examples of stromatolite horizons. You can see there's one here, and that's the edge of it going out into ordinary bedded rocks on the left. These stromatolite horizons are up to 10 meters thick and can be very laterally extensive, and they're comparable with younger reefs. And we quite often see biohermal deposits, you can see here this gentleman is standing on a stack of them. They're dipping off towards the right. They're kind of ellipsoidal in shape, and they're forming these discrete mounds with internal lamination and overturned margins. Nobody knows why they're quite that shape. So here's some of the details on the left-hand side of these of the internal structure of these biohermal mounds. And if we here we are again in East Greenland, and we can see hundreds of meters of carbonate rocks here. These are rocks that are from the Neo-Proterozoic era. That's the last few 100 million years of Precambrian time. And the stromatolites here form a significant proportion of these cliff-forming carbonates. So this sort of mating mountains out of microbes business, that's I had these sort of cliffs in mind when I wrote that title. And what we see in this slide is the rocks of equivalent age in Svalbard, in the Norwegian Arctic. There's a couple of formations here, and then this other one here is a stratigraphically slightly higher formation. And again, significant proportions of these beds are made of stromatolites. The lowest horizon here, this is called the Draken Formation, and it's got exquisitely preserved bacterial cells inside those rocks, and I'll show you those later in the talk. Now, on our expedition to Svalbard in 2011, Richard was a fourth-year undergraduate at the time, and you see he's holding this rock here. But Richard, do you know there's a rule, no personal specimens allowed on expeditions? And actually, I did snaffle it from him for the department when he got back, much to his chagrin. Anyway, this is his sample. Isn't it beautiful? But what do all those shapes mean? I mean, we can see all sorts of properties here. There's the width of each of these individual structures. We call them columns. We can look and see whether the layers overlap at the margins or not, how convex they are, what the shape of the margin is, but explaining them, that's a different matter. Now, the word stromatolite, it was translated from the German stromatolith, which was used by Kalkowski for the first time in 1908. So that word literally means layered rock. Over time, connotations of inorganic or organic origins have been attached to this word stromatolite, but stromatolites, as we understand them, are somewhere in the middle. Stromatolites are layered rocks created by sticky mats of microbes interacting with the environment. And specifically, they help sediment to accumulate, and mineral cements can precipitate inside them. And so they are often called organosedimentary structures, just like trace fossils. Now, here's a diagram of trace fossils. Now, you may be familiar with this sort of diagram, that trace fossils are burrows and borings, mostly in soft sediments. And we see a whole range of different ones here, from coastal through to the deep sea. And trace fossils have been given names, and they've been arranged in groups, like this Scolitos fascies here is a combination of several different types of trace fossil. There's Scolitos, this one's Diplocriterion, and there are others that have names. They're covered by the zoological code of taxonomy. And in many cases, they can be linked to a specific group of animals. For example, thalassinoides, which should be somewhere around here, is formed by a shrimp, and Scolitos is formed by a worm, and so on. So sometimes it's even a specific species of animal that forms them. So although trace fossils are indicative of animal behavior, they can be linked to specific animals or groups of animals. And so it's quite reasonable to give the trace fossils themselves names, even though they're a result of animal behavior as organic sedimentary structures. Now, does the same apply to stromatolites? Well, in the 1960s, Russian scientists systematically described stromatolites using a binomial nomenclature, effectively form genus, form species, form genera, form species were named. And one of the people that was involved in this is Mishi Semikartov, who came to Isla once. And this work was taken up in Australia by Malcolm Walter, who became very famous as a paleobiologist, Cass Gray, and also by Janine Sarfati, who I'll show you later, in France, and by others in India and China. So what they did was to take slabs of rocks and painstakingly kind of reconstruct them in 3D. Now, of course, these days you can do this by all kinds of automated methods. And obviously the IT is a different world, but everything was done by hand in those days. And they ended up with all of these different taxonomic groups of stromatolites. Now, in doing this, one thing you have to remember is that the total height of one of these columns isn't the same as the height above the surrounding substrate. So, for example, here, that level that's shown halfway up this box is the sediment surface. And so the structures would be poking up above the sediment surface by a certain distance, which will be much less than the total height of the column itself. And here you can see the first, on the right-hand side, the first time that sort of computers were used to generate these reconstructions. This is by a very clever guy called Hans Hofmann from Montreal in the late 60s, producing some of these reconstructions. And what Hans did was to summarize all the Russian literature. You can't read the individual names in here, but you just take it from me that there are lots and lots of different shapes that have got names assigned to them, which the Russians and others had developed. And so we had this quite involved taxonomy that existed, say primarily Russian-based, but also taken up by some experts in other countries. Now, if you then take that information and say, well, what does it tell us about evolution? And this is what it shows you, is that if you look at the number of different taxa, different named stromatolites over time, you see that there's a great abundance of them in the Proterozoic, particularly the later part of the Proterozoic. So the Proterozoic goes from 2.5 billion years up until the Cambrian. And so the Proterozoic was a time of great flourishing of different shapes according to this classification. And then it looks as though they're practically dying out at the end of the Precambrian. Now, this looks great, except that virtually nobody in the West accepted that this was valid. Well, I mean, some people in Australia, some people in France, but very, very few people did. And indeed, you know, in later years, people who were taxonomists were starting to work with people who were more environmentally focused and trying to reconcile their different views as to how to look at stromatolites. But it turns out that it's actually really difficult to reproduce all of the information that was used to develop this taxonomy. The idea of that you could determine evolutionary trends just didn't seem to be borne out by later work. So if there's a role instead for environment influencing, how could that actually happen? Well, stromatolites are much more difficult than trace fossils. They put on a good performance. Here they're showing off their arched layers, but many aspects of their formation, particularly what controls the shapes they make, are still tantalizingly unexplained, even well into the 21st century. Now, I started my research career in the mid-70s when there was a golden period of research about stromatolites and publication of a huge and wonderful book of this name in 1976. And yet the sedimentologist Kerry Kelts could write, "Stromatolites are a traditional mystery." In 1986, I gave a presentation at a workshop in Australia, the first time I'd been overseas for a conference. And I had a cartoon sketch, rather like the one on the left, saying, "Help, no one understands me." And it was a workshop all about, shall we have an international working group on stromatolites to see if we can understand them. And this led to Bob Byrne, who you can see at the upper right there, taking on the leadership of International Geological Correlation Program 261 on stromatolites. And this ended up with over 200 corresponding members in 34 countries. And this edition of the Stromatolite newsletter was all about this new project. Now let's wind back to the early days. We're going to go right back to the early 19th century. So in 1825, a guy called Steele published this nice engraving of some Cambrian structures from Saratoga Springs in New York State. And he thought they were giant concentric concretions like ooids. Now, later, James Hall studied the same outcrop, which is in a marine formation. And you can see from the different orientations of the outcrop, particularly this one here on the lower left, that they're certainly not spherical, but they're upwards growing mounds. Hall gave the first name to a stromatolite. He called them cryptozoan, which means hidden life. And then the famous American paleontologist Charles Walcott, who discovered the Burgess Shale fauna in 1909, he likened the cabbage-like cryptozoan structures to this sort of thing that you see in modern lakes. And these are lithified calcareous mounds, which are covered with a sticky mat of microbes. And this particular example from a lake is from Mexico. What Walcott did was to compare the modern lake structures with Precambrian stromatolites that he was studying in Montana. He found these flat laminated and domal ones, which he called collenia, and then the ones that form these more columnar structures, which he called cryptozoan. So he attributed both cryptozoan and collenia to blue-green algae. Today, we'd call collenia a domal stromatolite, and cryptozoan we'd call a columnar stromatolite. Now, I was lucky enough to see these classic rocks in Montana in 1989. And this photograph shows us a beautiful weathered surface of these mesoproterozoic stromatolites. They're between 1 and 2 billion years old, and they're from the belt supergroup. They have well-developed columns, that is, pillars of growth, here and here and here, in between which, in this case, you can make out debris. Can you see here, here, and here? There are pieces of carbonate sediment that have been broken off and incorporated in the interstices between the columns. And then you can have a look at the way that the stromatolite columns accrete. The accretion seems to be rhythmic, and you seem to have different kinds of layers. Another interesting thing is that the direction of upward growth changes, which is really weird. Why is that? Is it due to changes in currents, perhaps bringing in sediments, or is there any link to phototropism, which is the organisms responding to light? Lots of mysteries here, and the need for more studies of diverse modern examples is clear. So, enter a Great Britain. Maurice Black, there he is. So Maurice Black canoed the swamps of the Bahamas in an expedition in 1930, and he found modern stromatolites composed of carbonate sediments and organic mats, and so they're forming layers like you see in this image here. Now, later in his career, he made a different contribution to carbonate sedimentology. He was the guy that discovered coccoliths in the chalk and discovered that the chalk was largely made of these little tiny organisms. And that was, of course, by using a scanning electron microscope. And now I'm going to tell you an anecdote, so just relax slightly before we go back to the Bahamas. By the time I joined the staff at Cambridge in 1980, Maurice Black had been dead for 7 years. But there was another Black called Bill Black, who also worked on carbonate rocks, and Bill was on the staff. Now, Bill had a PhD, but official communications from the university in those old days referred to him as Mr. Black, not Dr. Black, because his PhD was not from Oxford or Cambridge. On the other hand, Maurice Black was called Dr. Black by everyone because he was so wise, but he never actually received his PhD. He objected to having to pay supervisory fees when his supervisor, O.T. Jones, took sabbatical leave one term, and he held on to that point of principle and never got his doctorate. Anyway, end of anecdote. Back to the Bahamas. In the Bahamas, the young Maurice Black found a variety of structures which he compared with ancient stromatolites. So Maurice noted several different types of growth morphologies. You can see these nice sketches here. Type C, for example, forming on the edges of upturned edges of desiccation cracks. Laminae were formed by layers of sediment coming in on tides or storms, or microbes whose activities vary diurnally. And we can see here some sketches of microbes that he found in the mats. He identified the species, these were all blue-green algae, as they were then known. Now, mats of that sort are found in many environments, regardless of climate. And we've got some here from Wales. This is from the salt marsh of the Lucker Estuary in South Wales. You see these, they're soft mats, they're mucilaginous, sticky stuff that's binding the sediment grains together. Now, those won't be preserved in the geological record because the organic matter will simply decay and you'll just have sand. But if you've got a situation where the sediment can be lithified, then structures like that can be preserved. I mentioned that Maurice Black had found blue-green algae, and so sedimentologists, in fact, were still calling them blue-green algae and were calling the structures algal stromatolites right up until the 1990s. But microbiologists in 1977 had decided that the absence of a nucleus in the cell of these structures made them more like bacteria than algae. That is, they are prokaryotes, not eukaryotes. Hence the new name, cyanobacteria, in other words, blue-green bacteria. So that's what I'll call them for the rest of the talk. Now, morphologically, these cyanobacteria can either be filamentous, as we see here, with a whole series of cells in a line, or they can be coccoid, with these individual more rounded cells. And in both cases, they're surrounded by this sort of mucilaginous material. In the case of the filamentous forms, it's like a sheath surrounding them. And this mucus is actually really rather important. It protects them from drying out, and it absorbs ultraviolet radiation, protecting the cells from damage. So that's pretty good, and it was good in the early Earth too, I should imagine. And these cyanobacteria practice aerobic photosynthesis, whereby carbon dioxide is consumed and oxygen is produced. Hence, the gas bubbles that you can see on the lower right image here on the surface of the map. Those are bubbles of oxygen produced by photosynthesis. Now, in the video that you can see later, you didn't see earlier, they make a big point of saying, think about the long-term significance of photosynthesis, because it's photosynthesis that eventually led to the Earth's surface becoming oxidizing. So let's set this in the context of Earth history. Now, the oldest stromatolites and the oldest fossils prokaryote cells are at about 3.4 gigayears, that is 3.4 billion years old. We'll see some examples of those later in the talk. Now, at that time, there is no evidence of any free oxygen in the atmosphere. So these things are busy producing oxygen, but what happens to that oxygen is it's used to oxidize reduced species like sulfides or ferrous iron to oxidized species like sulfate or ferric iron. And there isn't enough left over to accumulate oxygen in the atmosphere. That took a lot later. So you see, by 2.5 gigayears, there is evidence of oxygen building up in the atmosphere. We are actually seeing iron oxide, hematitic iron formations, for example, at that stage, and they increase in abundance to 2 billion years. So this great oxidation event is protracted buildup of oxygen in the atmosphere, still to quite low levels at that point, but nevertheless free oxygen was accumulating. And then after that, we're getting the eukaryote organisms appearing in the fossil record. That is, organisms with a nucleus in their cell. Oxygen levels stay low until really quite late in the Proterozoic, after the snowball Earth glaciations. Now then, a great breakthrough happened in Precambrian paleontology when people identified for the first time fossil cells of microbes. And this happened in 1965. And there are two workers, Barkhorn, who you'll hear about later, and Tyler. And they were cutting thin sections from this deposit called the Gunflint Chert, which exposed on the banks of Lake Superior. You can see here we've got volcanic rocks, and then we're getting cherts, silicious rocks developing on the top of those volcanic rocks. So they're kind of ocean floor deposits. And then in those cherts and those silicious deposits, there are stromatolites growing. You can see the stromatolite columns here in this thin section. And then looking at them in higher magnification and zooming in, you can then see these preserved filamentous fossils, which are fossil cyanobacteria, the first ones that have ever been found in Precambrian rock. So that was like really, Pandora's box open then in terms of there actually being evidence of Precambrian life. And the nice thing here, of course, is that the microbial content here is showing us that there is that the analogy with modern microbial mats with blue-green with bacteria is actually a reasonable thing to have done. That's what stromatolites represent. So that was important. And then people were doing various studies of stromatolites and both in the modern and in the ancient. And you end up with this idea that they're made of different things. One thing is that sediment particles are trapped, and that's these round orange things here in the upper left diagram are sediment grains, and you can see them forming layers in this lower left image. And then we've got the possibility of calcium carbonate precipitates forming, and sometimes they form discrete layers. Sometimes they seem to be largely a cement. And then you also get the microbes themselves growing up, and that they can trap and bind the sediment particles. Precipitation can occur around them, although it's not actually shown in this diagram like that. And just very occasionally you might see them preserved, but that's not normal actually to find them preserved. So you can see there's a complex interaction between the microbes and different processes of sediment formation, both sediment grains and mineral precipitates. Now, you might have thought from those people that had given all these stromatolites names, and the actual taxa change over time, and so they thought there was a clear evidence of evolution. But actually, when we look at the preserved cells, it turns out that the evidence for evolution is very limited. There are many forms that are exactly the same now as they were in the Precambrian times, and both early and later Precambrian times. There are one or two things that change, which I'll talk about in a moment. Now, one of them is the advent of something called thrombolites. Now, I was a bit worried at first when Moira Jenkins said to me, I hope you're going to talk about thrombolites, because I wasn't, because I've never worked on thrombolites. But it turns out that thrombolites are well worth including in this talk. They're a type of microbial rock that emerges late in the Precambrian, quite near the Cambrian, in fact. Instead of being laminated, it's actually made of very fine-grained, that is micritic carbonate in a kind of clotted fabric. And you see these strange looking clotted structures here. And because of the clotting, they're called thrombolites. And in the upper left, you can see this oval thing in section. That's the section of a bioherm made of these things. So they're like stromatolites, but they're not laminated. They're made of microbes that instead precipitate clots, mineral clots. And here's a modern example that Bob Burns showed me in South Australia. And you can see that it's covered with this this greenish mat. It's a microbial mat, and the material itself, this is weird, you wouldn't have heard of this. This is actually made of monohydrocalcite. That's calcium carbonate with water locked into its structure. Anyway, it's a calcium carbonate mineral, and it's a kind of got a clotted structure to it. It's not laminated, and it forms this hard mineralized structure, and it's forming in a modern lake. So there are modern examples of these sorts of things. And it turns out when people look at the microbes in these kinds of structures, the microbial community in them is a lot more diverse than simply having cyanobacteria. There are lots of green algae, for example, and there are simple animals, forams, for example. So it's perhaps logical that this sort of structure with these more diverse microbes would develop later in the Precambrian rather than earlier on. So there is an evolutionary link there. Now, in the mid-1980s, there was someone called Linda Moore who was working on thrombolite in Lake Clifton in Western Australia. So as we'll see in a moment, I haven't been to Shark Bay, the most famous place, but I have been to Lake Clifton, and I took this little bit of a video. And hopefully you'll be able to see the water surface moving over there, and those structures, those mounds are under the water surface there. Sometimes the water level drops and they're exposed on the shores of the lake, as we can see on the lower left photograph. If they grow up to the water level, then they tend to develop a flat top, as you can see there. So Linda Moore was working on these thrombolites there, and she was interacting with Bob Byrne about this, and they came up with a really good idea, and that is to say, why don't we introduce a new category of structures which embrace the thrombolites and the stromatolites and anything else that is produced by microbes. Let's call them microbialites. And this term microbialites has really become quite widely used. So microbialites are formed under living benthic microbial communities. That is, complex ecological associations, photosynthetic prokaryotes, eukaryotes, typically algae, and then different sorts of bacteria. Bacteria that synthesize their own food, that is the chemautotrophs, and bacteria which metabolize existing organic matter, that is chemheterotrophs. And so these sort of structures developed. So modern ones tend to be like that. They've got these rather more complicated assemblages than one sees typically in the Precambrian. And so they've evolved from those prokaryotic ancestors that dominated the Precambrian stromatolite record. Now, when Byrne and Moore defined this microbialite term, then they tried to sort of show the different types of microbialite and how they might form in different ways. So they had this tetrahedron where each corner represents 100% of something. So trapping and binding of detrital sediment will be up here, and then different sorts of calcium carbonate precipitation at the bottom, either inorganic or biologically influenced, or in the case of skeletal, that will be like a coral forming. So microbialites generally are not near the skeletal corner, but there's somewhere on the left-hand side. And if you look at this triangle here, this has got trapping and binding, and it's got those two things. So this is actually the triangle that's on the left-hand side of this tetrahedron. And in that case, you can see where Lake Clifton sits. Lake Clifton hasn't got very much trapping and binding of sediment. It's mostly biologically influenced calcification. Whereas in Shark Bay, which we'll see in a moment, it's much more towards the sediment binding and inorganic calcium carbonate precipitation. So microbial is a good concept to have, reflecting the diversity of modern microbial structures. Now, let's go back to the geological history. We said that near the Cambrian, things were getting a bit more complicated because we were getting a wider variety of organisms in the microbial communities, in the mats. But then we see this tremendous loss of stromatolites by the beginning of the Cambrian. They're much rarer from the Cambrian onwards. Why is that? The leading hypothesis is that they were restricted by the evolution of animals who predated on them, such as these modern snails, these Cerithiid gastropods here. You can see them grazing on the surface, having a lovely time. In normal marine environments, therefore, stromatolites become rare because the mats get eaten. But in freshwater or hypersaline settings, there would be fewer predators and microbial mats could become established. So that's the idea. So when from the Cambrian onwards, the idea is that most stromatolite occurrences are in those sort of settings. And it's been suggested that something similar happens after major mass extinction events too. So again, this is for Moira, because Moira was very keen that I showed a thrombolite. I think she's seen the thrombolites in Purbeck. And the upper left photograph there, that shows a microbial structure, a thrombolite that grew around tree stumps. So the trees were flooded by a lake that became evaporitic. And under those conditions, the benthic microbial communities could thrive. On the upper left one, that shows you a thin section of a few layers within a stromatolite from sky. Actually, you can't tell it's a stromatolite from just the picture. It just could be any carbonate sediment, but it does form a columnar structure in hand specimen. So that's from the Jurassic, from the estuarine series in Sky. So it's a kind of freshwater assemblage. And then a really famous rock, the cotton marble, forming in the late Triassic. And some people would argue that it's immediately after a mass extinction, and that's why it develops. But anyway, it's got layers which are stromatolitic, and it's got these peculiar arborescent structures which appear to be thrombolites. And there are two sets of them forming there. So those are some famous Phanerozoic microbialites that we see in England. Sorry, England and Scotland, I beg your pardon. Now then, this photograph I showed you at the beginning, it's on the Isle of Islay, and this is 2021. But in 1973, I did my undergraduate mapping on Islay in the Hebrides. And this island has the best developed occurrences of stromatolites anywhere in the UK. They're all Neoproterozoic, that is late in the Proterozoic, late in the Precambrian, and nearly all of them are preserved as dolomite, which is the dominant shallow water carbonate rock at that time. They're known from many horizons, both below and above the famous Portascaig Tillite glaciogenic deposits. Now, in the course of my mapping project, I looked at some of the stromatolites that had recently been described above the Tillite in this paper on the left here by Spencer and Spencer, with these nice drawings, which include these ellipsoidal balls that we can call bioherms, rather like the patch reefs that corals make. Tony Spencer had then recently published his famous memoir on the Portascaig glacial deposits underneath these rocks we see here. And a new version of that memoir we're working on 50 years later. But back in 1974, I then wrote an undergraduate dissertation on stromatolites, a literature review. And then I started my PhD on the formation that contained these described examples. And what I thought was needed here was a sedimentological interpretation of the setting. Now, for example, here we see a domal stromatolite that built on fine-grained dolomite, and that formed in quiet water. And then the morphology changes, and at the top, these then become columnar, and the columns are separated by a medium grain sand, which has been moved around by tidal currents. So clearly there's an environmental change there, and therefore an environmental control on morphology in that case. And what I did was to reconstruct the sedimentary succession by unraveling what was found in numerous small fault blocks. So we've got this spectacular coastline in the north of Islay, and the geology was broken up into lots and lots of little fault segments, and I had to sort of look at each one and correct for all the offsets and work out what the original geometry was. And it turned out there were several different types of sediment, several different facies representing slightly different environments, and including stromatolite layers, which are shown as black. So there's a whole series of these different stromatolite horizons. And I developed a model, an environmental model, with lagoons and tidal flats and tide-dominated estuaries. Now, at that time, there were a couple of prevailing models for sedimentation that did not fit what I found. Now, one of these was the dominance of the Arabian Gulf shoreline deposits, the modern Arabian Gulf shoreline, as a model to explain ancient shallow water carbonates. Now, along the Arabian Gulf, you've got quite long, straight shorelines. And as you go to, you have a supratidal zone, which in places like Abu Dhabi has got evaporites in it, what's called a xamka or salt flat, an intertidal zone, and then a subtidal zone next to that. And then, of course, you can then go off into what carbonate sedimentologists call a ramp, a gently sloping deposit further offshore. And what tends to happen is, as the shoreline builds out, you get the shallower deposits building on top of the deeper deposits. So you get subtidal deposits, intertidal deposits, and supratidal deposits forming this shallowing upwards cycle. And in my youth then, everyone was always finding cyclic sediments everywhere they looked. Now, of course, there are many of these that exist, but the problem was that I'd never seemed to work on cyclic sediments. It doesn't seem to work for me. And in fact, in Islay, I could show statistically that they are not cyclic. They're actually stacked very largely randomly. And so we need a different model in that case. And by 1986, Brian Pratt and Noel James came up with a model, what they call the tidal flat and island model, which involved a coastline that was not straight, that was actually physiographically quite complicated, with, in this case, perhaps estuarine with lots and lots of shoals and topography that varied in two different directions. And that way you could get facies on top of each other, which weren't forming very clear cycles. So that kind of model suited what I was finding much better. Now, another issue, another kind of thing that I couldn't quite relate to was stemmed from the discoveries in Shark Bay. And it's a really interesting example of the bandwagon effect in geology. And this particular bandwagon is that stromatolites were intertidal. Now, I was told this in a third-year lecture, but by then I had been doing a lot of reading, and I told the lecturer, the marvelous Dick Aldridge, that he was wrong. So we'll explain why. Roy McGregor from St Andrews was working on the Islay stromatolites during my PhD. So that's one reason I didn't do as much on them as I would otherwise, because he was doing this work. And Roy also had the distinction of having been to Shark Bay in Western Australia, the most famous modern example. And here they are in the intertidal zone. And he'd actually taken one of these things. You wouldn't be allowed to do it now, but he'd taken it and he'd sawn it. And there was a sample like this in the St Andrews rock collection. So these stromatolites are forming along the coast of this place called Hamelin Pool. So Hamelin Pool is a hypersaline inlet of the sea, as you can see here in Western Australia. It's hypersaline because there's limited circulation of seawater in here, and it evaporates quite significantly with a salinity much higher than that of ordinary seawater. And that means you don't get the normal invertebrates there. and microbial mats are able to flourish much more. Now, the stromatolites here were first noticed by a guy called Phil Playford and his colleagues in the 1950s. And they tipped off a young student called Brian Logan about their potential. And Brian really took hold of the ball and he ran with it. And he published a key paper in 1961 documenting these extraordinary shaped structures in the intertidal zone. Classic cryptozoans, you'd say. And that was then followed in 1964 by a very widely cited paper, Logan, Rezac, and Ginsberg. Rezac and Ginsberg being two pretty famous carbonate sedimentologists. So in the 61 paper, you can see what he has to say. He says stromatolites are composed of detrital sediment, and they result from the interaction of sediment and sediment-binding algal mat and environmental factors. So very clearly, environment is the key control. This is worlds apart from the world of the Russian taxonomists. So basically, he's saying that there has to be sediment trapping, and the lithification is sort of optional. The other thing that he said was that they are diagnostically intertidal. The progressive early lithification probably makes an intertidal zone location mandatory for this structure. So that was it. It was in all the textbooks. Stromatolites are intertidal. Now, in the paper by Logan et al. 64, which is quite an interesting paper, there's lots of good stuff in it. They made some comparisons with Precambrian stromatolites, and they developed a complex classification with codes here that people were meant to use. Of course, people didn't use them because they're just too complicated. But they developed kind of a completely different way of looking at stromatolites in terms of environments, and using the Shark Bay intertidal setting as the preeminent model. So they doubled down on the intertidal interpretation. And so the Shark Bay intertidal model became a meme, although the word meme hadn't been invented then, but it became a meme. And so if an artist wanted to draw a picture of what life was like on the early Earth when it first appeared, well, they used Shark Bay, and they had these things sticking out of the water. That was their image of what it was like. Now it's a false image, as it turns out. But anyway, that's what it became. It became a meme. And this artist drew this, I'm sure, much later than the time when the intertidal hypothesis had been discredited. Now, we remember Phil Playford. He was the guy that first discovered the shark-based stromatolites in 1954. He published this article in Science in 1967, which showed that stromatolites built reefs in the Devonian of the Northern Territories of Australia. So a lot of the reef facies here were stromatolitic. Stromatolites grew down to 45 meters below sea level on the Four Reef Slope. Now, the question about why they're so abundant here is something funny about this setting. There are lots of stromatoporides there as well. But anyway, stromatolites were very extensive across the whole of this marine reef, clearly completely different from the intertidal setting in Shark Bay. But actually, people didn't really take any notice of this paper, even though it was published in Science, the top journal. Not many people cited it. That shows the power of the intertidal meme. Now, that same year, 1967, Paul Hoffman, who later became famous for Snowball Earth, he published this great paper about these elongated stromatolite domes from the Proterozoic of Canada. And what he was able to show was that you can see that how the elongation of these structures, shown by these rose diagrams, is fairly consistent from one location to another. And it coincides also with the orientation of current structures. In other words, there were currents, probably tidal currents, going backwards and forwards here, and the elongation of the stromatolites reflected that. So that very much agrees with the idea that there's a strong environmental control on these structures. So it's that way it's consonant with Shark Bay. But were these actually intertidal? But then people started discovering subtidal ones. And here we've got an example of one by Conrad Geberlein showing stromatolites growing in current swept areas of the subtidal zone between islands in Bermuda. And then here's another example. This is from the Bahamas. It's at 10 meters depth in a tidal channel between two islands in the Bahamas. And it's and these are actually strombolites, actually, rather than stromatolites. They're still microbial. So structures like this, and they're quite happily growing in subtidal settings. Now, in 1972, Phil Playford wrote a paper in a Belgian journal about the Devonian reefs, and he let slip in passing that stromatolites were widespread in the subtidal zone of Hamelin Pool, Shark Bay. Surprise, surprise, and described this more fully in the 1976 stromatolites book. So there they are. If you dive down in Shark Bay, there they are. So that structures are not exclusively intertidal at all. And it gets worse than that, because it turns out that the structures all grew in the subtidal zone. And they have different layers depending on exactly what the environment's like. Some of them are more thrombolytic, some of them are more stromatolitic. When they grow in, when the reason that they're in the intertidal zone as well today is because sea level has fallen over the last few thousand years, and they've been stranded in the intertidal zone, and the nice laminated structure has been destroyed by a different type of mat, which is more of a destructive mat that's growing on the top of the structures. So there we are. So the intertidal mean was completely wrong. And in fact, it turns out that you don't get structures with significant relief developing in intertidal environments at all. They only develop in subtidal environments. Now, Shark Bay is still the preeminent environment for studying, in which stromatolites are found today. It's a World Heritage Site. But of course, there is concern, as with so many of these sites, about various environmental pressures on it, just as there was in Lake Clifton, actually, by the way, where the water pollution is a big concern. So going back to the Canadian examples, like those I show, those elongated ones described by Hoffman, other people have worked on these sort of structures elsewhere. This is from the Belcher Islands in Arctic Canada, and you can see lots of different stromatolite forms in the subtidal zone. Sometimes they're elongated, whereas in the intertidal zone, they're just pretty flat, just forming small little pustular structures. They're not forming big domes at all. Now, if we return to the Scottish case study, my interpretation of the environment showed a complex of shoals, sandy shoals, channels, and emergent areas where it became desiccated, rather like Pratt and James's tidal flat and island model. And when you look at the stromatolites, there's a nice little ellipsoidal bioherm. It's then draped by sediment, and the relief diminishes upwards. Here's a stromatolite column here, you can see, and it's being draped by other sediments, and its relief actually is lost upwards. So when these things, so the relief is not continually growing, it's actually diminishing, particularly when there's exposure. And this is nicely shown in this example. This is actually from Greenland. And when I went to Greenland in 1985, I was amazed to find exactly the same passage assemblage as the Isla stromatolites in one particular formation in East Greenland. And this nice example here shows stromatolites colonizing clasts that have been produced by desiccation and erosion on a surface. So they're in the intertidal zone, and they're forming a mound structure because they're draping over a clast. But as you go upwards, the relief diminishes, and that's what happens in intertidal settings. But I was certainly wrong about another thing, though, and that is that in the Scottish rocks there were these nodules present, and they're made now of quartz and calcite and pyrite. And Roy McGregor from St Andrews said these are anhydrite nodules. They're just like the nodules you see in the Sabkha of the Persian Gulf. But of course, I didn't. I didn't like that idea, and I said, "Oh no, they're metamorphic. They're nothing to do with sedimentation at all. They're entirely secondary." But then, of course, in the Greenland rocks, which have never been metamorphosed, these same nodules occur, and they've got anhydrite inclusions inside the quartz replacements. So these Scottish ones were certainly originally anhydrite, although they formed in a subtidal lagoon rather than a supratidal setting. Now, in the late 1980s, I studied several stromatolite-bearing formations in my first research council grant, and it was all to do with how does the carbonate differ in stromatolites compared with other carbonates that are not stromatolites. And the answer was not as much as you might think. So one of the things that carbonate sedimentologists like to measure is the carbon isotope composition of a carbonate rock. You know, it varies over time and can be used for correlation in stratigraphy. And a more negative carbon isotope signature is typical of a more organic influence. But I found there was actually no difference at all between the stromatolites and other sediments that were not stromatolitic. One of the nice things you can do is to tell the difference between layers that are made of detrital grains and layers that are precipitated. Now, in these rocks, there is some quartz and feldspar in this rock as well as dolomite. And if you use cathodoluminescence, which is how old-fashioned television screens used to work, you bombard something with electrons, you get this nice glow under certain circumstances. And the dolomite is glowing here. You can see all the crystals, but the feldspar is showing up as these blue crystals here, these blue grains. So each of these little grains here, this is a silt-sized feldspar grain. So you can see that there is detritus in here, but there is also precipitate. So these kind of clotted areas here, these very, very dark areas, and the right-hand picture are where there's precipitation of carbon occurring. So we're seeing both of them together in this, as you might expect to see in very many stromatolites, in fact. Now, here's Andy Moore. I'm going to tell you another anecdote here, because I had a great collaboration with Andy in the late 90s. Here he's seen on a later visit to Isla. Now, Andy held chairs. He's just retired. He's held chairs in both biology and geology at Harvard, just like Stephen Jay Gould. And he's established himself as the foremost Precambrian paleobiologist with a very wide grasp of geochemistry too. And he wears his modesty lightly. Now, let me up, right, sorry. Andy is the successor of Elso Barkhorn, who found the gunflint microfossils. In the corner of Andy's office is a safe. Why is there a safe there? I asked Andy. Oh, Barkhorn had it for the moon rocks, was the response. Yes, Elsa Barkhorn was asked to check moon rock samples for signs of life when they came back from the moon landings. But why is the safe still there? I asked Andy. This was in 1988, I asked this question. It's waiting, he responded dryly, for Mars. And so in 2021, or 2022, sorry, maybe in a decade's time, those samples from Mars may arrive. So early in his career, Andy made quite a splash by examining black chert samples from Svalbard collected in an early expedition. They were full of exquisitely preserved microfossils, and there's some beautiful examples here. So this is a chert, an early silicification, and you see these beautiful filamentous structures and coccoid cells preserved inside this chert. So he found some in samples that had been collected years before, and then he had the chance to go up to Svalbard himself. In fact, both Andy and I independently on different expeditions to Svalbard looked at the particular formation, the Draken formation that I've shown you in a slide forming a cliff earlier in this lecture. So let's just show you what you see. And the preservation is just amazing in this formation. The formation itself formed in a whole series of different sub-environments. Some of them were supratidal, some of them were intertidal, some of them were in lagoons, subtidal. And they've got these very distinctive fascies. Everything is beautifully preserved in very, very fine-grained dolomite and silica. So in the supratidal zone, this is where you're getting these things called TP structures that form by active cementation and pushing aside the carbonate sediment. So that was the supratidal environment. And then in the intertidal environment, locally, you get this amazing thing preserved. This is something called Polybessurus bipartitum. And in the 1980s, it had been discovered in silicified in East Greenland in the Precambrian and described by a student of Andy Knoll. And then, blow me down, it was then discovered in the Bahamas in the modern tidal flat after it had been found in the Precambrian. Exactly the same creature, no evolution, same thing. Found in the modern tidal flat. So it's a diagnostic tidal flat organism forming these tubular structures growing upwards. And these particular pictures here, the Svalbard examples, we've got them preserved in dolomite, whereas the original Greenland ones were in silica. And then in the stromatolite, this is a stromatolite here with several layers. And in some of the layers, you get these very, very fine-grained precipitates, which we see here under ultraviolet fluorescence. They fluoresce quite brightly, showing there's organic matter in them. And these are clearly filamentous cyanobacteria, exquisitely preserved in dolomite. And this is incredibly rare in the Precambrian. You occasionally get cyanobacteria preserved in limestones, in the Carboniferous, for example. But in Precambrian stromatolites, it's incredibly rare to see them preserved for reasons that are quite tricky to work out. We wrote a paper about it once, but it's quite a tricky thing to get to the bottom of. And then in the subtidal environment, you're also getting some really interesting fossils. This is a chert that's formed by silicification of a subtidal mud, and it's got in it different types of plankton. You've got this big thing here. This is an acritarch. It's an organic walled microfossil, and then you've got these tiny little blobs. These blobs here, and these are what are called vase or vase-shaped microfossils that are simple unicelled animals. So they're accumulating in this lagoon. And then this amazing picture here in the lower left, this shows a whole series of sedimentary layers, and there's these little tiny filaments going up through them. They're just preserved as slightly coarser dolomite crystals, and these are the right size and shape to be green algae growing up, baffling the sediment. So they're not, for some reason, they're not distinctively creating a kind of wriggly layer like a stromatolite would. They're just ordinary sediment layers that have been baffled by green algae. So we're getting a real window into a really diverse microbiota here in these stromatolites. And then one other example of Precambrian stromatolites I was working on in the late 80s, I'll mention here, is one I've already talked about when I've talked about Snowball Earth. And that is that in association with the glacial deposits of Snowball Earth, we find these glacial lake deposits. And the glacial lake deposits here consist of layers of limestone, which are shown in white, and they're calcareous precipitates, interlaminated with layers of sediment that have been dropped from icebergs floating in the lake. And in places, there are areas of which were cavities that are now filled with cement, which represent places where the mat has lifted off the bed of the lake, being billowed upwards by gas. And you actually see this in modern Antarctic lakes. So these are deposits from a modern Antarctic lake with white calcium carbonate layers, clastic sediment shown as this darker stuff. And here we see these are mats, microbial mats. You can see the oxygen bubbles on them forming by photosynthesis, and they're forming these kind of cone state structures, which is because of the generation of gas that's lifting them off the substrate. Now, when people first found these Antarctic structures, or these microbial mats with of this shape, they said, "Oh, they're conical. There are Precambrian stromatolites that are conical. They're called conophyton, and they're very common. Maybe this is how they form." But this was nonsense, because conophyton is solid. It doesn't have gas bubbles underneath it. It really represents layers that are very steeply conical. And in the subtidal environment, in many Precambrian environments that are subtidal, where limestones are accumulating, you get these things rather like rocket cones. Like imagine a field of rocket cones on the seabed. That's what these conophyton are. So for some reason that isn't yet fully discovered, if you get a quiet environment offshore, the microbial mat forms these conical shapes, and they're formed largely by precipitation. There's very little sediment inside them. Now, the only time I've had to see structures of this sort was in the late 80s. I had an opportunity to visit West Africa and Mauritania, this amazing country of Mauritania, which of course has suffered a lot from human activity. But I mean, in terms of its natural environment, is incredibly beautiful. So we're on the western fringes of the Sahara. We're crossing a bit of sandy desert here, and we're ending up in the rocky deserts of the Attar district of Mauritania. And you can see here on the right, these are domal stromatolites forming distinct layers. There's a whole series of these stromatolite horizons forming this mountain here. And this is Janine Sarfati. Janine was born in Algeria, and she's kind of lived and worked in the desert for a lot of her career, although she's been based in Montpellier in France for later in her career. And here she is on a visit to Islay in Scotland. So the stromatolites here in the Attar group, there's lots of different horizons, and there are lots of these conical structures that you can see on this log here in the center. And these conical stromatolites look a bit like this. You can see them forming these dark gray structures. And then in between them, there is this quite complex sediments and cements. So these are typical offshore sediments. But what else is you get something even more bizarre associated with these things. And that is a stromatolite which Janine has called jacutophyton. And this really is a stromatolite that deserves a name because it's so distinctive and so bizarre. So what we've got is there's the kind of rocket cone of the conophyton core, and then here it is on the right hand side too. There's the conophyton core, and then around the core then develop what look like branches, but you can see them in plan view here. They're concentric around the structure. They're like petals surrounding the conical core. This is and they're shown here. They actually kind of grow a bit more upwards than is apparent from this particular the diagram. But this is the idea. You've got a cone, and then you're getting these petals growing off it. And a few decades later, Linda Kay and her group looked at these things in more detail, because I was completely baffled by these things when I was there. But Linda's worked at them and worked out that the reason that these things develop isn't environmental control. It's all to do with the change in sea level. And that when you're getting shallow water conditions and more turbulent conditions, you then start to get these petals growing, and they're associated with the breakage of material falling off these conophytons. Whereas if the conophytons stay in deep water, they just stay as that very simple shape. So over the last, now I've emphasized a lot that we don't really understand the shapes of these things. And over the last 20 years, many people have grappled with mathematical growth models to help explain stromatolite morphology. So these are basically algorithms to simulate growth. And here's an example by Grotzinger and Knoll where they do this. And they have a formula to show a certain amount of growth, and they allow sediment to migrate towards depressions. So that so the high areas continuing growing up, whereas the stromatolite growth is inhibited in the depressions because sediments accumulating there to a greater level. And then here's an example where you're getting cement growth, and you can simulate cement growth in a similar way. So you can actually build up a mathematical model and create growth. But what it's very difficult to do is to really come up with a number of the really distinctive aspects of the stromatolite column morphology. The degree of convexity, for example, the steepness of the slopes is really very difficult. Now, this makes life particularly difficult then if you go back to very old rocks, where the question is, had life even evolved at all by the time this rock formed? And therefore, are these structures biogenic, or could they have been formed by some abiotic mechanism like you would in a computer model? So if we go back to 3.4 billion years ago, and this is from a paper by Alwood et al. In Nature in 2006, they found all these sorts of morphologies in stromatolites from what's called the Strelley Chert in Western Australia, some of the oldest sedimentary rocks on Earth. And are these structures biogenic? And they convinced themselves that they were. And they got drawings here of these structures. They reckon there's such a wide range of particular and distinctive morphologies that there really is hard to explain them unless they're biotic. And then they also have specific things here, like they're looking at the shapes and angles of growth here and saying that they think that the axes of the stromatolites are aligned to the paleo-vertical, because these were formed on a slope, and therefore they're illustrating phototropism. In other words, the mat was growing towards life. So there we got 3.4 billion years ago, arguing very strongly that structures that look like stromatolites really are stromatolites. And they're also, after great argument, some people have demonstrated that there were also microbial cells in rocks of that age. So that's when we can be sure that we've got microbial life around on Earth. And then another example of the limits of stromatolite formation. When is a stromatolite not a stromatolite? When is it something entirely inorganic? Is the example of manganese nodules. Now, you may have heard manganese nodules are in the news again now. They used to be in the news decades ago. They've come back into the news because they are full of precious, well, they're full of metals that we want. Manganese nodules are found in the deep ocean. For example, the Clarion-Clipperton zone of the North Equatorial Pacific. They're found in the deep ocean where sedimentation is minimal, and they grow very slowly as manganese oxide bodies up to a few centimeters across. And they contain metals like nickel, cobalt, copper, molybdenum, titanium, lithium, and rare earth elements. And so for decades, there's been talk of mining them. Legally, they are the common heritage of humankind, which means either they belong to whoever gets there first or hands off. Anyway, this is currently a looming threat to deep marine ecosystems if one of the oceanic nations goes ahead and starts mining them. Well, in cross-section, manganese nodules show this kind of columnar structure. And numerous studies have shown that they also contain bacteria capable of redox reactions, particularly oxidation and reduction of manganese, for example. Well, back in 1973, the famous Belgian stromatolite worker Claude Monti proposed that manganese nodules were bacterial stromatolites. And more recently, a Japanese group, who'd obviously never heard of Monti, independently proposed the same thing, and noting that the structures had kind of fractal properties. But the question is, can they really be bacterial stromatolites? Because these structures only grow up to 10 millimeters in a million years. So can something that grows at 10 millimeters in a million years actually be microbial? So as you see, I'm not giving you many answers this evening. Well, a promising way forward for the future to understand the genesis of these different shapes would be by laboratory studies. And here's an example where people have taken mats from a Brazilian lagoon and they've actually cultured them very successfully in the laboratory. And that allows them to determine the biogeochemical responses of the mat and its morphology. These kinds of studies are at an early stage, but experimental studies are definitely a brilliant way to go for the future, because all the other approaches have proved remarkably difficult to get the kind of breakthroughs that we might want. Now, here's the sawn specimen that the lovely people in Callow End have sawn. Here's the stromatolite. It's crazy, isn't it? It's not simple. Here you can see some columnar structures, several of these. There's a close-up of one of them. These columnar forms are preserved in dolomite rock. This is from Isla, of course, Perseus Farm, that was heated to 450 degrees during metamorphism. That is the rock, not Perseus Farm. But the rock and the stromatolites remain intact. They're crazily individual and unnameable, as outlandish and inscrutable as ever a stromatolite was. And finally, here's a couple of book suggestions. This book is called Stromatolites, Ancient, Beautiful, and Earth-Altering. I guess that applies to some of us too, and particularly the ancient. And this one, Blame It on the Stromatolites, Real-World Creative Thinking for Young Adults. Stromatolites started it off, after all. And that's all for me. Thank you very much.